Hi, good evening, folks. This is Bolton from Committee of 100. Thank you all for attending. As we wait for more of our attendees to trickle in, we'll start shortly in a couple of minutes. Thank you again.
Welcome, everybody, to the Committee of 100's discussion with our very well-respected C100 member, especially during this time, Dr. David Ho, who is the CEO and Scientific Director of the Aaron Diamond AIDS Research Center here in New York City. Dr. Ho was with us earlier in the pandemic to let us know where we are at. He is going to let us know again where we are at. The title of our Fireside chat with Dr. Ho is Reopening with Dr. David Ho. So first off, Dr. Ho, how are you?
I am well. Good evening, Richard and fellow members, and good afternoon or good morning, wherever you are.
It is great to see you, and your work has been well documented since we have spoken. But why don't we start with this? How would you say things have changed? If you remember our conversation from a long time ago, you've had many, but what has changed since then from your point of view?
Well, the epidemic has changed in that it has gotten worse globally speaking, and certainly gotten worse in many regions of the United States. On the other hand, scientific progress has been made. There's been considerable progress on the development of vaccines and antibodies, and that is certainly good news. And we could get into those topics a bit more as we proceed.
You bet. So for those who are just joining us, thank you for being here for the conversation. Just a quick run of show, if you will. We will be speaking with Dr. Ho for a little bit, about 10 minutes, 15 minutes. He's going to now give us an introduction to discuss what is new with his Columbia team. And then we'll get into some questions and answers at the end. So you can put your questions in the Q&A section at the bottom, and we will try to get right to them. Dr. Ho, open it up for us in terms of what your team has been doing in that same time frame that I was just discussing with you.
Yes, our biggest project has been focused on the discovery of antibodies that are capable of neutralizing SARS-CoV-2 or COVID-19. And I could get into that a bit more since that paper was just published last week in Nature. But we have also been working on the discovery of small molecule drugs that could potentially block the protease of this virus. And protease, as I discussed previously, is an enzyme that's involved in cutting the virus proteins from large chunks to small pieces. And we have identified a number of small molecule drugs that could intercalate into that protease and thereby block its function. However, these are not at a point where they are considered drugs at this point, or drug candidates at this point. We are continuing to go on those small molecules to optimize their potency, and hopefully down the line we could turn them into drugs. Another project that we are working on is actually focused on coming up with a test that is different from all the tests that are out there right now. We hear about antibody tests that will tell you whether someone's exposed to the virus in the past, or the so-called PCR test that looks for the viral nucleic acid. But that latter test, the PCR test, is in fact too sensitive. And it picks up traces amount of the virus, even though there's no longer any infectious virus around. So we're designing a test now to measure one infectious unit. And if it's below an infectious unit, one infectious unit, then that person is probably not infectable or infectious and not capable of transmitting the virus. So we hope to develop that test in the coming weeks and apply it to help reopening society. But our major project is this antibody project.
You said a couple of weeks. Is it because you've been working on it already for many months? It certainly takes longer than that for you to create such a test, I imagine.
No, we've been developing the reagents necessary to build a very sensitive, it's actually called an antigen test. And that antigen test we've been refining over the past couple months. And we're now at a point where we're reasonably pleased with it to proceed to test clinical material. So yes, it's not something just in the works from the last week or two. It's been in the works for some time.
For some time. You know, you were mentioning your article that was talking about antibodies. And for those who were unable to see it, it's published in Nature, and we'll also put the link down in the chat section. I see we've already put that there. Dr. Ho, I'll read the title: Neutralizing Antibodies Isolated from COVID-19 Patients May Suppress Virus. This was the headline from the posting on Columbia University's Irving Medical Center site. Tell us, summarize what is in that article, and it just published July 22nd.
Well, let me tell you the purpose of the study first, and that is we want to isolate antibodies that are extremely powerful in inactivating the coronavirus. And the reason we wanted to do that is that such antibodies could be used to treat or prevent infection. For example, if you had a really powerful antibody, you could give such an antibody as an injection to people who are recently infected but are not severely ill yet. And the hope there is to stop the progression of the disease to respiratory failure, etc. On the other hand, it could also be used to prevent infection, particularly among those individuals who may not have a robust immune response to vaccines. We know vaccines are coming along nicely, but vaccines generally perform less well in elderly individuals and in people with underlying conditions or those on chemotherapy or immunosuppressive therapy. So the antibodies could be a substitute and antibodies serving in the role of what we call passive vaccination. So those are the goals of our antibody discovery effort. So what we've done is to evaluate a bunch of patients who presented to the Columbia Medical Center and study them carefully to see who carry the most antibodies against COVID-19 in their blood. And from the 5 top cases that we selected, we were able to draw their blood, isolate what we call B cells, and specifically B cells that recognize the surface protein of COVID-19. And then basically using modern single cell cloning methods, get the antibodies out, get the antibody genes, and then reconstruct the antibody. And we, in doing so, came up with about 250 antibodies. And in particular, there were 61 that were very good at neutralizing the virus. And we obviously zeroed in on those that are most potent. And that we focused on 9 that had exquisite potency. That is very low concentrations of the antibodies capable of neutralizing the virus.
And the hit ratio that you had, you said 61, right? Is that pretty good?
Yeah, we like to think so. I think basically we pulled out a total of 252 monoclonal antibodies. About half of them actually specifically recognize the surface protein of COVID-19. And then of those, about half are neutralizing the virus. So that's pretty good in our opinion. And then the 9 with great potency, that is pretty unique because many other groups are doing this. And generally, each group pulls out a couple potent ones.
Well, why are you having such a high hit rate or a higher hit rate? What do you attribute that to?
We attribute it to, I would say, good technical expertise, but in part also to patient selection. I think we, unlike many other groups, they just study any case that showed up. We selected from among 40 patients the top 5, and I think that played a very important role.
And what am I? This is the article. What are these 2 drawings?
Okay, well, this is a very important picture for me to get into the next point. These are two pictures of the spike protein, which is the center part of the molecule, and you see shades of green and orange there. But the blue parts are the antibodies, and on the left, we have identified a new class of antibodies that attack the orange brownish region of the molecule, which we call N-terminal domain. And then on the right side, you see a picture of antibodies attacking sort of the green portion, that the receptor binding domain. So in addition to the potency of the antibodies we isolated, we have found antibodies directed to different sites of this spike protein.
Interesting.
And that's important because it allows us to form rationally antibody cocktails. And cocktails are important because they will prevent the virus from escaping from a single antibody.
Yeah. Now tell me this, Dr. Ho, is 1 great, 2 fantastic? Do you need 5? What's like the number of different attack zones, if you will? You've identified 2 in these pictures.
Yes, I think overall in the current study that we reported, we found 3 different regions where the antibodies attack the virus. But in terms of what is needed, it's probably a bit risky to go with 1 because this virus is an RNA virus that's capable of mutating and escaping. So the general consensus in the field is we probably need 2. 3 might be an overkill, but we certainly would like to go with 2. And the fact that we discovered a second and a third class is really helpful for constructing antibody cocktails.
Yeah, as my grandma used to say, good on you. So 2, it's basically attacking from 2 sides. It's cornering the virus, I guess, and just kind of getting at it. Is that why 2 is the magic number? I'm probably oversimplifying.
Right. I think basically if you attack 2 sites, the virus would have to simultaneously mutate at those 2 sites.
Got it.
From a probability calculation, that would make it much less likely to occur. And so that's basically the strategy that was worked out years ago for preventing HIV escape from a cocktail of drugs.
What is this picture?
That's the same thing. It's again, the bottom on the left, the bottom part is really the spike protein, and in cyan and dark blue are the antibodies basically binding to the top of this molecule.
Yeah.
And in this particular case, preventing the virus from binding to the receptor. So it shuts the door for the virus to enter a cell.
What's the green again?
Green is part of the spike, and the region is what we call RBD, as written there in green, the receptor binding domain.
And on this picture here,
that's the same molecule rotated 90 degrees forward. So we're looking at
the top,
yes.
top down here. Yep. Okay. Really, really cool. And in real life, how big is this?
You wouldn't be able to see it. And this is visualized through what we call cryo-electron microscopy.
Easy for you to say.
It's amplified many fold.
So we can't even say how big it is. Yeah, this is so teeny. Here's the electron microscopy 3D reconstruction. There you go. Antibody cocktails you discussed. Talk about the implication for vaccines, Dr. Ho.
Well, so these antibodies were induced when people were infected with the virus. So the immune system saw the virus and mounted these responses fairly quickly within a few weeks of infection. Now, we also looked at the genes that encode these antibodies. And it turns out that these genes did not have to go through an elaborate evolution before the antibody was generated. And without getting into the technical details, the gene sequences for these antibodies are very close to what we call germline. That is, the immune system did not have to go through a whole elaborate process to come up with these powerful antibodies. That bodes well for the prospect of a vaccine protecting. So the immune system is naturally ready to see coronaviruses. Now, we have to explain why that is, but maybe through evolution of mammals, this has been encountered, and the immune system of humans, like other species, are already primed to see this. So it is a good sign that vaccines could protect.
So instead of a vaccine being given to a patient, it's not a 100-pound lift for our immune systems to get to these antibodies. It's a 20-pound lift, if you will. You're saying it's easier.
Yes.
Okay.
Exactly.
You know, tell us about your team. This is a picture of your lab. Who is this, and what are the things we're seeing in this picture of your lab?
That's my colleague, Manoj Nair, and he's one of the few individuals who work with the infectious virus, and hence he's dressed up in the way you see it there with a respirator, since this is a respiratory pathogen. And he's in our biosafety level 3 containment facility. And when he works with the virus, he will only do so within those cabinets. He would stick his arms into the cabinet.
Right here? This one on the right here?
You see two of them there, side by side.
Yeah, this one here too, right.
And the cabinets, they're laminar flow so that materials do not come out of the cabinet. And then they are vented out through a filter. So
out the top here somewhere.
Yes, exactly.
Yeah. And these are essential, I imagine, and you have two of them in here. And the suit that he's wearing is completely enclosed. It looks like there's supply air coming up to the head here. Is that right?
That's correct. That's so that his exposure is minimized as much as we can.
Yeah, and what's back here, Doctor? Oh, all these. Is this one of those mixers back here?
I'm not sure I could see that all that clearly.
Okay.
Probably supplies on a shelf.
Yeah, that's okay. I will discontinue our eye check so that we can see you now. You know, one of the good news pieces, and again, folks, if you got questions, you're just joining us, welcome to Reopening with Dr. David Ho, put on by the Committee of 100. It's our second conversation. We're going to get to that topic, by the way, but I want to talk about some good news with Dr. Ho. He's already shared with us just now what he has discovered with his researchers. And how many again do you have on your team, Dr. Ho?
How many people?
Yeah.
Well, our whole institute right now has about 40, 50 scientists. But I have two individuals who work with the infectious virus, and we have a whole team that's involved in the antibody work. That's about a dozen people or so.
That's fantastic. That is great. One of the good pieces of news as of maybe this week, as we count, is what the New Yorker, I think, has counted 27 separate vaccine efforts. Is that about right, Dr. Ho?
Well, there are a lot more vaccine candidates than that. I would say maybe several weeks ago, when I took a close look at the number of vaccines being developed, there were 17 that were already in clinical trials. And you've been reading about many of those from Moderna, Pfizer, Novavax, and several from mainland China as well. But there are close to 150 still in preclinical development.
Wow. Wow.
So the vaccine effort is enormous. And this is unprecedented in scale and also in speed, in that many of the vaccine candidates went from the lab to the clinic in the course of just a few months. And
wow.
A bit unbelievable, actually. I
think what the number was 27 vaccines that have demonstrated some efficacy in animals, which is the positive is the positive headline. Does that sound about right though?
Roughly speaking, I think it's right.
Okay. What about therapeutics?
I think other than antibodies, the drugs are slow in coming, and that's because you need to discover these small molecule hits, and then they tend to be low potency, and you need to go through iterative optimization, and that takes longer. And then since those are small chemicals, you also need to be very careful about safety testing in animal species. So that takes a while. If you don't have any toxicity in the first days or first week, you still need to see if you have any late sequelae of importance. Mm-hmm. And so it takes more time to develop the small molecule chemicals. The antibodies, on the other hand, are moving along very rapidly, not just those from us. And I should have said earlier, we're not the first to come up with powerful antibodies against SARS-CoV-2. We just feel that we have come up with some of the very best and some of the most diverse panel antibodies.
Is it because most of these antibodies came from those in what was a former hotspot where we had so many severe cases? We're talking about New York.
Yes, so the antibodies have been generated by a number of academic groups as well as pharmaceutical companies. Regeneron and Lilly in this country are leading the way, no doubt. And there are a number of companies in China that are developing monoclonal antibodies. But of late, in the past several months, academic groups like ours have come up with a whole bunch more. And so now they're all being put into the pipeline for development, including ours.
Back to your team, where was your team from? What countries do they hail from? What towns do they hail from?
Let's see, Shanghai, Chengdu, Xiamen, some town in India that I can't recall. Of course, different places in the U.S. Myself from Taichung, from all over the world, basically.
That is amazing. And how did you select your team? That's doing this team that is the strike team for COVID.
This team basically is a team that's been together for a number of years, working on HIV, specifically on antibodies that could neutralize HIV. So we have a lot of virology expertise and antibody expertise, which we then turn to COVID-19 starting in March or so.
That's great. And you were also mentioning the town you were hailing from. Um, we have one question in here that I wanted to share with you since we did get to that point from Shu Ren. She wants to know, tell us about your career. And, you know, here you are working in the United States. This is Committee of 100, U.S.-China relations is the focus. How did you come about, in a nutshell, coming here and then becoming what you are and being a leader in this space?
Well, you know, I early on in my medical career, I developed an interest in infectious diseases, and it was in the early 1980s when, you know, fortuitously, I saw some of the early cases of what we now know as HIV/AIDS, and developed an interest in that condition, even though it was quite esoteric at the time. But to me, it was interesting in the sense that it was a mysterious illness that caused the destruction of the immune system, and that was transmissible. And I knew, even as a younger scientist physician, I knew that was scientifically important, and that piqued my interest, and I pursued it without realizing early on that it would ultimately turn into one of the most significant pandemic in human history. And so I worked my way in that field, and you know, made my share of contributions. And then you know, as I'm getting older and beginning to think about retirement, but then COVID came along, and
We need you. We need you.
those plans have changed for now.
We need you. We need you, Dr. Ho. What was it like being Asian in your profession?
Well, I think in the, you know, when I went to medical school, in a class of 140, we had four Asian students, Asian American students. Nowadays is quite different. Particularly, you know, in schools like Columbia, you know, you see a huge cadre of East Asian and South Asian students. So that's no longer all that unique, and it's good because I felt a bit lonely going through medical school, but things are quite different nowadays.
I'd say. I want to get to a couple more questions, and then I'll go back to COVID and reopening specifically, if these questions don't specifically address those. And I'll start from Carolyn. Carolyn Chin writes this: I am immunosuppressed due to transplants, 72. Are any of the vaccines likely to be useful for those like me? Any of those over 27 that are doing well right now?
Well, obviously, we don't know with certainty whether someone like Carolyn will respond to the vaccine. But if past history is any indication, someone on immunosuppression is not likely to mount a robust immune response to any vaccine. And that, by that I mean active vaccine, the kind that mimic the virus given to the person hoping to stimulate the immune system to mount a novel immune response. That's not very likely to occur at a level that would achieve protection. And this is why part of the reason we went for these antibodies, because now we could identify the most potent antibodies, produce them in outside the body in factories, and then give it back to individuals such as Carolyn, so that one injection could protect, say, for a period of 3 months. And in fact, the antibody could be engineered to persist longer, so that perhaps it could go out to 6 months.
That is amazing. That is great stuff. Carolyn, see, you got all the answers here with Dr. Ho, and that is specifically addressing his latest publishing of the article. Another question: are the antibodies collected from all currently known strands, or the mutated versions of COVID-19, or both?
I think that's a pretty sophisticated question. There are obviously the virus is evolving, and many sequences have been determined, and we are looking at the major variants. And you may have heard that the virus that swept through Europe and hit the east coast of the US has a unique mutation at position 614 on the spike. And that there's some evidence to suggest that's more transmissible, but we have looked at that particular variant and know that it's very susceptible to the antibodies that we have come up with. And we're looking at other major variants to be sure that the antibodies could neutralize those as well.
Lori asks this, if not today or August, would you say that people should have reasonable comfort returning to offices?
I think that depends on where you are. That's clearly not the case if you live and work in Florida or Texas or Arizona or certain parts of California. I think New York, we have opened up to a large extent, but I would still say that despite that, in New York City still has a couple hundred cases per day, and that's, you know, in Beijing they would be hitting the panic button with that kind of numbers, and in Hong Kong likewise. So we have, you know, different tolerance levels. So I think everybody, despite, you know, New York doing much better, everyone still needs to be quite vigilant in wearing masks, in social distancing, and in maintaining good hygiene.
You said you were mentioning different panic buttons. What's the right panic button then? Should we be hitting the panic button?
I think obviously in about half to two-thirds of the country, we should be hitting the panic button. I mean, the caseload of 70,000 per day is unacceptable for a country like ours, which is strong and rich, and yet our response is like a developing country. And that is because I think we don't have a national strategy. We don't have the leadership that is willing to commit to solving this problem. You know, it's left up to the states, and when we need a unified response. And, you know, what is the right answer? Well, there are success stories out there. China is fairly normal now, although people still wear masks. South Korea and a number of other Asian countries are doing relatively well. So it could be done, but we refuse to follow the formula that has attained success elsewhere, and that's because we value individual freedom so much that we forget that with that freedom comes with responsibility, especially when it comes to public health.
And when you look at the curve today, and I'm going to pull that up very quickly here, going to the New York Times very quickly. This is the coronavirus death topping 150 as of today, according to their numbers, and it varies depending on which publication. But what do you see here, Dr. Ho? Is this going to go back up to what we had over here earlier on in terms of the deaths per day?
Yes, I think it's gonna climb and hopefully plateau, but I don't know exactly at what level. But if the infection rate is any indication, we expect death to lag behind by several weeks to a month. So we know that situation in much of the Sun Belt states have been pretty bad since June. And I suspect the will follow. We know the hospitalization has followed, and we know what the state of the bed shortage and all that in places like Texas and Florida. So surely the death rate will rise. The only good news in all that is that the demographics of the infection have changed somewhat, in that a higher percentage of younger folks are being infected now, and hopefully that results in lower mortality rates.
Look at these numbers here. I know they vary depending on whose count it is, but in general, the trend here, Dr. Ho, of new deaths up 53% on a 14-day trending line. Yet the 14-day trending line for new cases is only up by 5%. That used to be the big number, right? And the smaller number used to be the deaths. Expected, unfortunately, just based on the way the virus works. Do you think we've hit the second plateau right now? The number of cases seem to be leveling off a little. What's your thought?
I think we cannot be looking at the U.S. as a single entity. We have so many places experiencing different epidemics. So the tri-state area where I reside resembles Western Europe. We went through a big outbreak. It's come down to a relatively low level. But if you look at a place like Louisiana, it got hit in March and it was brought under control, and now it's in its second wave. If you look at California, it got hit and it was plateaued for a while and just kept going up in recent weeks. In Florida, there was a delay, and then it peaked after May. Well, it continued to rise after May. It's not. Hopefully, they have reached their peak and will crest and come down, but time will tell. But basically, if you have this kind of infection numbers, a smaller percentage will need hospitalization, and an even smaller percentage will result in severe disease, and some of those will die. And you could see that's going to lead to a delay, especially in death, because many of the patients struggle for weeks before dying. So the death numbers will lag behind by a good 4 to 6 weeks sometimes.
What do we see on this? What do you see in this map here, Dr. Ho, this heat map?
Well, it tells you where the problem areas are, right?
Right.
Reflected by the darker shade of red. And it's what I've been talking about. It's the Sun Belt and a little bit on the west. California was the first to lock down, and they have, I think the governor had made many of the right decisions, but California also has a lot of migrant workers living in closed spaces, and that has exacerbated the situation there.
With the therapeutics, with the treatments that we've got right now, how much smarter are we at battling COVID-19? If I were to get it today and I have to go to the hospital, roughly speaking, not an exact number here, Dr. Ho, but how much better have we gotten at battling that disease, that virus, if I were to get it today and I had to go to the hospital?
Well, as opposed to 3 months ago, I think we know with greater certainty that remdesivir is a drug that is helpful, although it's far short of a magic pill. It's slightly beneficial. I think the studies show that, and so that's helpful. We also have learned in the intervening period that dexamethasone, which is a steroid, could cut the inflammation and be helpful based on a pretty large study from the UK. And so we have more definitively shown that there are two agents that are helpful. But we also have lots of confusion out there. I think to the mainstream medical community, hydroxychloroquine is dead. It's clearly not useful and at times harmful. but we still have a doctor who believes in witchcraft, and our president who clearly doesn't have a license to practice medicine, giving out misinformation.
Adeline Ma asks this: when a patient receives a vaccine and produces his own or her own antibodies against the virus, how are those different than the ones that you're producing in your lab?
Well, I think that's being characterized now by the vaccine developers. So they could measure in the blood for the same activities against the virus and so-called neutralizing antibody activity. And it seems that they are, the responses in the vaccinees are reasonably comparable to some of the antibody levels seen in infected individuals. So that too is a good sign for vaccine development.
When you're looking at the vaccines, what are some of the key? This is a question from Xinjin Zhao, and the question is, what are the key enablers for the unprecedented speed that we have all of these, well, at least in, there's 24 at least that are looking pretty good? What made this happen?
Well, that's because there was so much new technology development to address certain epidemic diseases out there that were tough. Number one on that list is HIV. Number two is malaria. Number three is TB. And all those technologies that were developed to address those pathogens were immediately applied to COVID-19. Once the viral sequences were available, all the companies already had certain platform technologies, and they simply cut and pasted the genes of the spike protein into those platform technology and just proceeded. Now, one thing to bear in mind is that while the prospects look good right now for vaccine, many of those strategies that we're talking about, whether it's the so-called RNA strategy, DNA strategy, particle strategy, many of them don't have a lot of clinical experience. And so we don't know whether there are any long-term complications associated with those. And we also don't know whether they will elicit an immune response that is durable, right? If it's only there for a few months, that is not good enough. And then would require booster shots and perhaps repeated booster shots. And that takes away the value of a single shot vaccine. So we need to bear in mind, and so Moderna with its RNA vaccine, there are no RNA vaccines out there today. So one might ask, are there long-term complications? Well, we don't know. So would you like to be one of the early recipients of such a vaccine? So people have mixed feelings about that.
A surgeon has his favorite tool or her favorite tool. What is your favorite tool in your lab as you battle this beast? You talked about a lot of new technologies. What is the favorite new technology this time that you didn't have when you were doing HIV/AIDS?
Well, 5 years ago, we didn't have in our lab these single cell techniques. So in the process of discovering these antibodies, I mentioned earlier, we could go to a patient, isolate the B cells, and then hone in on those B cells that could bind to the spike protein. And now we could take such spike protein bound B cells individually and put them in droplets and sequence the antibody gene from the single droplet.
Oh, yeah.
And then pull that out and then reconstruct the antibody. That kind of technology is only recently developed. And
what's it called?
Well, it's a machine that's called 10x Genomics that we use to do that.
10x Genomics. Everybody on this gathering is now doing a Google search, I'm sure, of what that is. Another question for you here. What question are we not asking that we should be asking?
Well, I think there are still many very important questions that are not answered from a scientific and medical perspective. Why is it that so many people are asymptomatic and so many are mildly symptomatic? And why is it that some people, even relatively young, succumb to this disease? There's clearly a genetic component that we haven't fully defined. We know a little bit about minor contribution from the ABO blood group, and we know that there's a spot on a certain chromosome that's involved in inflammation that may predispose someone to a worse outcome. But we need to understand the genetics more fully. And the other aspect is, there are two phases to this disease. Early on, it's a upper respiratory infection, just like flu and many other common cold viruses. And most people just recover, but a small subset go on to have a second phase that is full of various complications. It's obviously what people have called cytokine storm, lots of inflammation, but there's much more than that. It's not just pneumonia, it's multi-organ failure. It's as if the clotting cascade and a complement cascade in the body are triggered. And exactly how that occurs, we don't know. So if we could understand that process better, we could develop therapeutics to intervene and help to save those patients with severe disease.
Alice Young asks this, practically speaking, what kind and level of masks should seniors be wearing? Can you discuss microbial masks as well as silicone masks? Are they the trend? And also, another question, just to add to all of that for you, is discuss herd immunity if you can, in addition to all of that. Those are two questions that we have.
Yeah, I'm no mask expert. We wear masks, and when in very, very risky situations, as you show in the picture, when one of my colleagues is working with the virus, obviously we wear, fully protective mask with respirator, and so that's much safer. But I think the so-called N95 mask is what we would wear if we're seeing patients who are infected, and that's very protective of the frontline healthcare worker, along with other things. But that mask is crucial. But in terms of the effect of the mask in protecting the public, it is largely to confine the secretion of the infected person. That is its major contribution. And there you don't necessarily, I think a surgical mask or a cloth mask would already do 90+% of the job. And the reason that universal masking is important is that if you ask everyone to do so, you surely will confine the spread of the virus from the infected person without any stigma or discrimination involved. And also we know, unlike many other diseases, this one has a asymptomatic carrier state being so prevalent. And therefore, you really need universal masking. You can't just say, okay, you're infected, you need to wear a mask, because you don't know who's infected. And so that's the key. But in terms of, there are obviously many types of masks, which I cannot speak about with expertise. In terms of herd immunity, I think, in general, as a population deals with the infectious disease year after year, or sometimes decade after decade, more and more of the population would have been exposed and would have an immune response. And the immunity of the population then could potentially protect a person who's not been exposed. But that requires something like 60-70% of the population to have immunity in order to be effective for that unexposed person. And we simply cannot build up herd immunity fast enough. If we do so, we're going to see a huge surge in death that our healthcare system cannot absorb. And so, when we say flatten the curve, we want to flatten the curve so that the healthcare system could absorb the surge, and delay the cases as
Right.
long as possible.
Greg Lee asks this: many think in the winter we'll have another wave, and is it possible that people will catch both the flu and COVID-19? And it's been talked about before. How bad is that double hit, if you will? And then related to all that, Dr. Ho, where do you see this current spike going, and for how long, and what are the scenarios after this spike?
Respiratory viruses generally do spread more easily in the winter season because people are indoor. They're generally closer to one another, and the conditions are more favorable for the virus to survive in the environment. In the summer months, with people outdoor and with greater sun exposure and the UV kills the virus, it's believed that respiratory viruses would generally slow down, and that is the case with common cold coronaviruses. They tend to decline substantially in the summer months. But we're not seeing that with COVID-19 right now. I think it's spreading in many warm and sunny places like Florida. But that also means when winter comes, we could experience worse. And that's something that many of us are quite concerned about, especially the fact that the U.S. epidemic is still very, very bad. And if we don't quickly in the month of August and September bring it under control, the situation will be tragic in my view.
What does this say about this virus, the fact that it is uncharacteristically, if you will, surviving during this period, during the heat, during summer?
It tells us that this virus grows very well and grows to extremely high levels, and that because of the high amount of the virus in the secretions, it's much more easy to transmit. In the laboratory, we could see the virus grow at enormous levels, levels that are way beyond what HIV could do. So it's a bit frightening.
When the vaccines come out, what would you suggest to people that are on this gathering today? Should we take it immediately? Should we wait? For those who don't want to take it, is that, and what do you also say to them?
I think it would have to depend on the specifics of the vaccine, in terms of its protective efficacy. Is it 100%? Is it 99%? Is it 90%? Or is it 60%? So the performance of the vaccine needs to be factored in. Is it one shot? Is it multiple shots? And then, what platform, how much safety information we have had by the time you need to make that decision. What are your personal risk factors? Are you a young man like yourself, Richard, with no medical conditions, or are you a 75-year-old with hypertension and diabetes? This would all one's calculation. So I think I'm reluctant to provide a general advice. I think it needs to be evaluated based on so, what we know and what one's risk factors are.
That was Chris Alberti's question. A quick follow-on to that, Dr. Ho, from Janice Wan. She wants to know, would the vaccine have more efficacy depending on region, race, or anything related to that? Age, I think, is one of the points you just brought up.
Age and underlying medical conditions and therapies could affect the response rate. In general, we haven't seen dramatic differences between different races of people. I think age and underlying condition are the key factors.
This is from, I want to go to this question here because I'm trying to get to as many as we can here, Dr. Ho, in the remaining minutes we have. This will be the last one. How concerned should people be in certain regions of the country, and should they leave those areas, or should they now, because the duration is so long? What would be the suggestion there?
Well,
I know, not an easy one.
Not an easy one. I think it depends on one's personal circumstances. When New York was undergoing its rapid spread of COVID-19, some people left, but many chose to stay and stay home, basically. And I think if one were a particularly high-risk person in Florida, I would tend to avoid going out and basically stay home and minimize the risk that way. But it all, again, has to depend on the circumstances of each individual.
Those who have recovered from COVID-19, this is a question from Michael Shea, have they, I'll read it again. How have patients who have recovered from COVID-19 been infected or reinfected? I'm sorry. Have you seen that happen before? And how long do the antibodies work?
That's a very important question that we don't have a definitive answer right now. There are anecdotes of reinfection or relapses. Most of those are not well documented. In animal experiments, we haven't seen reinfection. That is, the immunity induced by the initial infection seems to protect against virus challenge subsequently. So there's no clear proof of reinfection in humans at this point. In terms of persistence of the antibodies or immunity in general, typically following a virus infection, you would have a peak in the antibody response. And weeks after that, you would see some degree of waning of the antibody levels. But keep in mind, our immune system has memory. So we have these memory B cells that are primed to recognize that particular pathogen. And should the virus come back and invade that particular person, those B cells will kick into action in a matter of a day or two and pump out lots of antibodies. So my personal belief, based on my understanding of viruses and the immune system, is that that's going to confer a great degree of protection. Even if the antibody levels wane with time, the memory B cells are there and they will kick into action.
Owen Lee asks, what do we know about the infection and transmission by children at this point? Schools reopening, right, as they go through that, and younger ages.
Yeah, I think, COVID-19 is teaching us something different about respiratory viruses. Typically, we know from our personal experiences that kids catch flu and many other common cold viruses rather readily and spread it around easily. But COVID-19 seems to infect children less. And however, they are clearly infectable, and they could clearly be the source of transmission to their parents, to their family members. It seems that, in particular, kids under 10 don't have as severe outcome from this virus as older kids or adults. Teenagers are, I think, more or less like adults with respect to COVID-19.
Why is this the dynamic in young children? Do we know yet?
We don't know for sure. There's a lot of speculation about the fact that in the respiratory tree, from the mouth and nose all the way down to the lungs, the cells express a molecule called ACE2, and that is the receptor for the virus. And there's some evidence to suggest that younger children have lower levels of ACE2. So there are fewer gateways for the virus to penetrate into a cell. That's not definitively proven, but that's one speculation.
Could someone who is infected but non-symptomatic become lifelong carriers of the virus? Helen Gong.
Not likely. These viruses are not persistent viruses that could insert itself into the host like HIV or like hepatitis B, or like some of the herpes viruses. But it could persist for a number of weeks. And there are case reports where the virus is still detectable maybe 2 months out. But I would say most of those are just viruses being detected by a very sensitive test called PCR, which is could pick up a few molecules of the viral RNA, and yet those numbers do not constitute one infectious unit of the virus.
We're getting close to the end here. We're going to finish in about 2 or 3 minutes here. Yulin Shu asks this: antibody testing right now and how it's changed since you and I were talking about in our last conversation. You showed me a little sample. I actually took it, came up negative. I had to because I had come back from California. I also took the nasal swab in addition to that, came up negative. And this again, because I happened to be in that state when it became quarantine again. This is not too long ago. So Yulin is asking about taking this antibody test or a test before going back to work, and is it a safe practice that that some organizations may be using before they bring people back to work?
Well, I think antibody testing is a good way to understand what percentage of our population in a given area is already infected or have been infected, right? Antibodies generally would take a few weeks to develop, so that indicates past exposure. It doesn't indicate current infection or not. But that's still useful information. And it might be useful to a given individual to know that he or she had the virus before and may have immunity. As I said earlier, the question of immunity is yet to be formally proven, although I think it's likely. We do develop immunity to most viruses. Why should this one be different? And so that might be useful. I mean, when you took your test, I mean, in some way, you probably hope you already had the antibody, right? And I know a lot of people who took the test and were a little dismayed with the negative result. And so I would rather have antibodies than not have antibodies.
Whoever thought we would want to say that out loud, right? Final question, which is the title of our conversation. We've been talking about it, but reopening. What is the good state model that you've seen so far that you think is a good way to approach it?
Well, I think, you know, what has occurred in New York State is exemplary. I mean, the initial outbreak was bad, and you could look back and said a number of things could have been done better. But in terms of then bringing that enormous outbreak under much better control is uh, is a good example for the rest of the nation to follow. And yet, you know, we see so many states wanting to repeat the same mistakes, not wanting to follow the rules that were laid down, that were proven to be successful. And it's not just New York State, right? You could look at Italy and France and many Western European countries, as well as South Korea, mainland China, Taiwan. Uh, Lots of success stories out there, and yet, you know, there's so many people who are so arrogant uh, to not want to follow good examples.
Dr. David Ho, for our fireside chat, reopening with Dr. David Ho, hosted by the Committee of 100. Dr. Ho, thank you. Thank you again. And I appreciate, along with all the other members and everybody here, all your work. We are cheering you on. We are cheering on everybody in your team. And thank you for educating us, making us a little smarter today. Thanks again, Dr. Ho.
Thank you, Richard, and thank the committee for putting this together.
And thank you, all of you out there that are watching and have been with us, and for your questions. We try to get to most of them. For now, until the next fireside chat with Dr. Ho, have a great evening, a great day, and a great week ahead wherever you're at. Thank you, Dr. Ho. Thank you, Committee of 100, and thanks to all who are here. Bye-bye.